Hi, everyone. I'm Shannon Wyman, the Scientific Communications Manager at Keystone Symposia, and I'll be hosting this Meet the Editors Roundtable today. I have with me today editors from Cell Press, Nature, and EMBO journals to discuss the state of the field, exciting new directions in cancer stem research, as well as tips on how to publish in these top-tier journals. Before we get started, I'll go through some notes on how we will run the session today. First, please keep your camera and microphone off during this first portion of the event. We will start with a 30-minute discussion with our panel and then open up the discussion for audience questions. You may submit your questions at any time into the Zoom chat. During the audience Q&A, I will select questions from the Zoom chat and ask the person who submitted the question to turn on their video and microphone to ask the speakers your question live. After asking your question, please turn off your microphone once again. And please leave the Zoom chat clear for submitting questions. Okay, now I think we are ready to welcome our editors. Our editors could please join me on screen. Great. We'll get started today by having our editors tell us a bit about their journal and their role there. Sarah Hamilton, would you like to start us off?
Sure. So my name is Sarah Hamilton. I'm the editor-in-chief of Cell Reports Medicine. So this is a new broad scope gold open access journal at Cell Press. It's focused on translational and clinical work. So this is one of two medical journals that have been launched at Cell Press in the last year. So the second journal is Med, who published their first issue in December. Cell Reports Medicine started publishing last spring, so we celebrated our 1-year anniversary in April. So we're a team of 4.5 professional editors, half because one of our editors is also at Cancer Cell. We're located in the US, Europe, and China. Our team is growing, so we're in the process of recruiting another editor right now. In terms of scope, so cancer obviously is an important part of it, but we're really interested in papers in all areas of medicine. And again, with an emphasis on translational or clinical work. So, work that helps us understand health, disease, medicine, and ultimately those that are going to help drive therapies forward.
Great. Daniel, would you like to go next?
Sure. Good morning, everyone. I'm Daniel Klimek. I'm senior editor at EMBO Journal. I'm based in Heidelberg in Germany, but obviously we're a global journal, broad scope. We're probably more on the basic side of disease biology, interested in molecular, cellular, and also tissue biology of cancer. We are a long-standing venue, so this year we celebrate our 40th anniversary. So it's kind of a classic and the oldest of the EMBO Press family of journals. We're not for profit and more community-oriented, and we want to offer a fair, fast, and informed peer review process, which is transparent, and I can talk about that, which is, and should serve the community in its best sense. Thank you.
Great. And Sarah Seaton-Rogers.
Yes. Hi, everyone. I'm Sarah Seaton-Rogers. I am an American based in London. I work for the journal Nature Reviews Cancer, where I have been for the past 15 years. I'm now the chief editor, and I've been chief editor for 7 years. And I guess we're in between the two other journals in that we're celebrating our 20th anniversary later this year. So the Nature Reviews series is 21 years old, and we were part of the second group that launched. So basically, our scope is basically in the name. We cover review type articles only. We don't publish original research papers. So what I'm looking for in a conference like this is a little bit different than what the other editors are looking for. And it's all on cancer biology. So we're not super clinical. We tend to focus on the cancer biology side of things. We have another journal called Nature Reviews Clinical Oncology, and they publish more on the therapy of patients and the direct clinical implications. Obviously, there's some overlap with translational work, and sometimes it's difficult. There's a gray area in between, but we've tried to define our scope mostly as being cancer biology, and we're quite strict on that now to avoid overlap with the other journals around. So I think that's pretty much an introduction to the journal. Our team is very small. We have only 2.5 people at the moment. We should have 3. We have a part-time person while someone is on maternity leave. So in a few months, we'll have 3 again, but we're a very small group of editors who runs this entire process of publishing all the reviews each year. So it's a lot of work, but very rewarding. So thank you.
Wow, great. It'll be really interesting to hear the difference between the reviews publication and the primary research articles editors.
Yes, it's
great.
Definitely.
Well, to kick off our discussion, let's hear from each of you. What are the most exciting advances you've heard so far at the meeting or looking forward to hearing about tomorrow? And what are the new directions in the field? jump in, whoever wants to go.
I can start maybe.
Yeah, you can start since I just spoke, and I'll go next.
Yeah. Well, I will just kick it off by saying it's amazing how it should be really encouraging times for the field, because there's so many. It's really a nice crossroads of advances in the methodologies, right? So we heard about. For a couple of years, we heard here at the meeting about heterogeneity and how to resolve it, and how important, particularly for the cancer stem cell field, how to disentangle some of these subpopulations, and there all the single cell sequencing methodologies like transcriptome, ATAC-seq, maybe also proteomics, imaging, spatially resolved methodologies. I think that's really to advance us further. And then this comes across with new and innovative in vitro 3D models. So we heard a lot about organoids of almost any tissue at this point. And of course now, I guess it's an exciting phase to pull this into the cancer context and ask, the tumor organoid, as we heard today from Michael Shen and others, what can we learn from it? Can we look at the human context? Can we learn something about the longitudinal aspects of cancer progression? So I think that's really aspects I think are exciting.
Yeah, it is really exciting to see the organoid applications grow, as well as a research tool, as well as a translational tool. Sarah Seaton-Rogers, I think.
Yes, I was actually going to say something very similar to Daniel, and I think that I cover everything in cancer biology, so I dip in and out of different conferences on different topics, but I've been to a lot of cancer stem cell meetings over the 15 years I've been in the journal. And I do think that it's kind of changing finally, that there are these technologies available that are going to allow us to address some of the questions that may have hindered therapeutic targeting of cancer stem cells effectively in the past. So I'm really hopeful that some of this single cell and spatial analysis and the organoids and all that, just as Daniel said, will work and help us with that. I was also going to say that I thought it was really interesting to hear more about this terms of how they interact, the cancer stem cells or tumor initiating cells, with everything else that's in the tumor microenvironment, be it the other cells there, the immune cells, the fibroblasts, the extracellular matrix, that sort of thing. I think we heard some really interesting talks on both days so far about that, and all of these systems, things that we have and the models that we have are going to better disentangle that as well. So that's also very interesting.
Yeah, I think the single cell analysis has become really powerful lately in, distinguishing those different groups and what they're doing together and separately.
Right.
Sarah Hamilton?
Sure. I mean, I'd be reiterating what Sarah and Daniel said. It's been a really interesting mix of, methodology, basic biology, like deprofiling, some clinical work, although some of that will come tomorrow. So it is kind of interesting just to see the field develop over looking at some of the talks here, compared to a decade ago, where people were really looking at the role of extracellular matrix and just how much more sophisticated the methods are now. So really starting to understand it. And I'll be curious, looking forward to seeing kind of how these culture techniques can be used, of course, for the basic biology, and that's a lot of what we've seen so far at the meeting. But also, for screening platforms, all the rest of it, drugs, trying to predict responses, et cetera. And then there was some work looking again, as Daniel and Sarah mentioned, about tumor heterogeneity, and so there were some interesting talks on looking at how that heterogeneity can help you understand responses. So really, why some patients are responding versus not. And I'm really looking forward to tomorrow's session on clinical translation. So
great. So to dig into a little more detail, what do you have your eye out for to publish in upcoming issues of the journal? More basic discoveries, translational, clinical insights? Does it have to include both? If you can give our audience a bit of a insight into what kinds of stories you're looking to publish. We'll go to reverse order this time.
Okay, so starting, sure, happy to. Yeah, and that would be pointing to what I have thought is interesting. So as I said, our journal is focused more on the translational and clinical work. Our sister journal, Cell Reports, is really more focused on the basic biology. And then we have another Cell Reports family member that just launched Cell Reports Methods that would be interested very much in the methodology. That's not to say we're not interested in the methodology. So as I said, these culture techniques are pretty interesting. We would be looking for how they're leveraged clinically. So again, if you're using them to predict responses or for drug screening, something like that. We have been publishing papers, that are deep profiling of tumors. So some of that work would be relevant and is of interest. Again, there's a lot you can do with that kind of profiling and that analysis. You can get a lot of different insight from it. Ours would be more on the clinical end. So for example, analysis that's stratifying patients, or again, predicting responses, or trying to understand why some patients are responding versus not. So we would be looking more for that clinical or therapeutic take-home message from these analyses, but still very, very relevant. And then again, we're looking for work that is on the clinical end of this. So targeting these populations as a therapy itself, or as a way to improve clinical outcomes, overcome resistance, et cetera. So the more clinical papers would definitely be of interest.
Great. Sarah?
Yes, so I guess I'm up next in the middle. That's good. So I mean, I'm totally different, as I alluded to earlier, in terms of that we don't publish the original research. So we don't come to these meetings so much to look for stories that are, of interest in terms of unpublished data. But in terms of what we publish, we're looking at review type articles. And when I say that, I mean reviews and perspectives. And perspectives are essentially opinion articles. Then those are written by researchers, primarily invited by the editors. So although we do take some proposals, which I'll discuss when we talk more a little bit about tips for submitting to our journal. Basically, we're looking for themes, right? So we come to the conference and we look for ideas and threads that run through maybe more than one talk. I mean, it's pretty rare that a talk is a review in and of itself. Sometimes it is, but we're really looking for something that's a little bit different in terms of having a unique angle or being an up-and-coming topic that is based on unpublished data that once published may change the way we think about things. We're trying to really do something a bit different in terms of providing a unique synthesis of the data that's out there. It needs to be different from stuff we've published previously, which is always a hurdle, and other journals too. There are a lot of review articles out there published by my own journal, by our own publishing house, and many others as well. So trying to find a unique angle and something different is always what we're trying to find. In addition, meet new authors. That's another goal of us coming to these conferences, to look at the different speakers who are here and people we may not know already, and networking the same as everyone else is doing. So I think that kind of sums up the primary things we publish. We also maybe less well known to many of the audience is that we publish some shorter pieces that are also written by the science community and the researchers, and they are basically commentary type articles, or what we call worldview articles, which are somebody's unique perspective, very short, of interest to the field. So we've started doing more of these in recent years, and they cover a lot of things that are of interest to the scientific research community, and maybe more of a sort of science and society angle, issues facing early career scientists as they're launching their careers, or issues related to research funding, issues related to bullying and harassment and diversity and things like that. So we're trying to do more pieces like that. So that's another angle that we're looking for, which sometimes comes up in the scientific sessions as well.
Great. And Daniel.
Yeah, so I should mention actually that I also cover review articles in front half, but I'll talk about the primary stuff here. Yeah, I think for the primary articles, I'd like to highlight two things. Yesterday we had this splendid talk from Barbara LeSavage on the stiffness and how will it impact on the epithelial, the PDAC and in the pancreas, but also Valviva's work on mammary gland biology. So how does rigidity of the tissue change along the progression of the cancer, and how does it impact on the seeding, metastasizing, disseminating cells? And so this context, I think, is really only recently coming up across the different cancer entities, and we would be really interested in the signal transduction, so mechanotransduction machineries affecting this, and probably how to tackle this in cancer. So that's the one area I'm really interested in. And the other is like I think in the cancer stem cell field, still the long-standing question is that, like a chicken and egg thing. So who is instructive? So is it like the stem cell will kind of hijack existing tissue stem cell niches and kind of exploit them and just drive in as into a parking lot? Or will it be more like the stem cell really instructively imposing a rearrangement of the niche? And I think recently we've learned about systemic metabolites affecting tumor microenvironment, and also at the interface between the cancer cells and the immune cells and the systemic cues. I think that's really exciting, and yeah, that's an area we're certainly interested in.
Great. So I'm sure we have plenty of scientists in our audience looking to publish their work, if not now, within the next year or so. What would your best piece of advice be for them in submitting their work to you to increase their chances of it getting accepted? And that can be either something they should do or maybe something they should not do when they submit their work or reach out to you. Anyone can jump in on this.
I suppose I can go first on this one since I haven't yet. And obviously what we're looking at is quite different. And I said already, alluded to this, that I would discuss this when I mentioned proposals. And so what I mean by that is if researchers have ideas, if they're interested in writing a review type article of any size or shape or whatever they're interested in, commentary type articles as well, I'm happy to look at a proposal submitted. We have a whole system on our manuscript tracking system online where people can submit a proposal. And I mean, the main issue is that we receive a lot and we can only take so many. And as I mentioned before, we have to think about overlap, but really we're looking for, as I said before, something unique. So tell us how it's unique. So provide us with some rationale for why this is timely, why this is unique, why it's different from what's out there, how it's based on research that's been published in the last, I don't know, 2 to 3 years, perhaps, something interesting, and, explain to me why it's interesting for a broad audience of cancer researchers, who are going to read it, and not something that's field specific. I think that's really, really the best piece of advice I can give.
Great.
Whoever. Yeah, maybe I can. Ahead. Yeah, I will just mention 2 things. So the first would be, I suggest to think about, to have a good grasp of what a variety of different journals interested in. It's because the stories you'd like to tell and to share with the community will come in different flavors, and maybe you need different venues to kind of accommodate those best. And so I suggest to have a good grasp, to go to a few journals you're interested in, and then really look what type of stories do they do. You can look at the content, you can look at the referee reports, even in the editorial exchange, to see what's the type of discussions, and you'll find out that's the type of story which might work well for EMBO Journal or another journal. And then and also at which stage what's needed to hit this level, basically. And because for our journal, for instance, we would encourage submission of rather mature pieces, so that means that if we take them along, that we can publish them within a good half year. So not within 2 or 2.5 years, but rather in a concise major one major revision. So that will mean you would have to submit at a rather complete stage already. Have a good grasp of the journal, and then the next would be approach the journal, right? So be very proactive in basically making a point for your interest, for your research, for your perspective, a unique perspective, as Sarah just said, from your point of view, and be visible not to your peers, but also to the journals, and that can go via many channels these days. This can be via the one-to-one session at the Keystone. This can be via just mailing to the editor. I think any journal is really interested in getting input, and so we'll always be attentive to these.
Great. Yeah, I think this comes up a lot. Knowing the scope and what the journal covers and not submitting to the wrong place is a key part of that submission process.
I wouldn't say wrong. I mean, if there's no wrong there. It's just that. It might be that I mean, usually journals try to respond very fastly, and then in a few days you should get an answer if there's really a mismatch. But I think,
best fit then. And Sarah Hamilton.
Yeah, I'd be reiterating what others said. Really pay attention to scope of journal, of course. If you're not sure if it's appropriate for us, then talk to us. So that's the big thing is really take advantage of the different avenues in which you can interact with the journal. So pre-submission inquiries, a lot of journals have those. Turnaround is very quick. Or just email. Cell Press editors, you can find all of our email addresses online. We're pretty happy to talk to you. If you have a more specific question about the journal or the paper. Daniel made a really good point about thinking about who you want your audience to be. You know, a translational paper really can go in a basic biology journal. It could go in a clinical journal. Think about who you really want to see the work. Where are you trying to drive the field forward, and kind of pick accordingly. And then again, in the vein of talking to us, use things like your cover letter to the best advantage. Like, this is really candid place that you can talk to us about your submission. It's at Cell Press anyway, it's for the editors only. And so, use it to put your paper in the context of the field. Don't just reiterate your abstract or anything like that. You can be candid about competition, things like that. So mostly I would say really take advantage of the various ways in which you can communicate with your editor, with the journal. Write your paper clearly, accurately, really try and avoid a lot of hyperbole. Most papers are in advance of some kind, and we're going to recognize that. So really talk about your work, clearly, concisely, accurately.
Great. Yeah, and I really do want to stress that to our audience. I know editors can seem like daunting behind the scenes sort of people that are kind of deciding on the fate of your work, but as you can see, they're really approachable, and they're actually there to help you, and they're a great resource to help direct your paper. I want to touch on what you just said a bit about the cover letter, and dive a little more into that. What is the difference between a cover letter and an abstract? And what are the types of things that should go into a good cover letter?
Sure, I'm happy to go since I brought it up.
Sure.
I mean, an abstract is really more of a concise overview, a couple sentences about the background, what your goal was, what you did in broad strokes, not really getting into the minutia, and then what you found, with a sentence about what this might mean. And so there's only so much you can cover there because you're trying to touch on a lot of different points without going into too much detail. The cover letter really, it's really just, I'd say, expanding on some of those things. Where I find it really useful is where you provide your thoughts on where your work fits in the context of the broader field, how you think it's moving a field forward, where you might not be able to get into as much detail on that in the abstract. I do see people that just cut and paste the abstract, and that's really a missed opportunity. I know cover letters are they're one more thing that you have to write. It's one more barrier to submission, but it's something I read on absolutely every submission, and so I think it's worth it. And then again, you can in cover letters be more candid about what's going on in the field, or if you have specific competition and you need to let your editor know or have them be aware of that. So you can be more candid there than you would obviously be in the abstract.
Mm-hmm. Mm-hmm. Daniel, do you have anything to add?
Not much to be added. I'd say the difference is, as Sarah just said, that the audience, right? Or that you with the cover letter, you address the editorial team primarily. With the abstract, you address all the field basically. So it's really different. And yes, in the cover letter, you can be a bit more informal. You should it should be complementary to the main manuscript, and in some ways, make a different point about why there's a gap in the knowledge of the field and why your story is kind of coming along. And you can tell a bit more about that, maybe also competing work or complementary work. So a bit of the background of the story and how you came about spending 5-8 years or whatever on that piece of work.
Great.
But I'd say it's a bit overrated. So in our view, we always try to go in depth, to read in depth into the main manuscript, and then at the end, I might take a few minutes to skim over the cover letter as well to get that information. But I think the core convincing piece should be the actual manuscript.
Got it. And Sarah Seaton Rogers, did you have something to add?
I don't really have a lot to add to that. I mean, for us, cover letters, yes, they're important when we get pre-submission inquiries of proposals and things in a way, and I prefer it when people don't just copy paste the abstract, which we do see as well. But at the same time, when, you know, if we've invited a submission and people send a cover letter, it's kind of not necessary because we've already asked them to do that for us. Mm-hmm. But I mean, I've obviously read a lot of research papers over the years, so I kind of understand how the cover letter works anyway for the research paper. And I feel that, and I think a lot of people take it as the opportunity to sell their research, and that, but without being, you know, too hyperbolic, as Sarah said. You know, you don't want to oversell it, but to explain why it really is filling this gap and why it's interesting and why we should care. And that's useful. But yeah, the research, yeah, as Daniel said, should sell itself, hopefully. But it can't hurt to put a little bit of extra spin on it, the same way you would if you were applying for a job. You know, you send your CV with the whole list of everything you've ever done, and then you also write a cover letter, right? So that's your opportunity to explain why you fit in that place, which be it a new job or a new journal.
Mm-hmm. Great. Yeah, and I know we've talked to a lot of editors at various eSymposia meetings, and it seems like a personal preference as to if the cover letter is really important to one editor or just kind of an extra for another. So I guess it also depends on who you get.
Yeah.
Great. I wanted to talk a little bit about the pre-submission inquiry. What would be an appropriate example of when someone would submit a pre-submission inquiry versus just submitting the paper, or more informally contacting you versus an inquiry?
I could give an example maybe. So, so per se, I think a pre-submission inquiry is not what we're 1,000%. You know, what we would prefer always. I mean, I think it's a starting the dialogue, but we can, like in the busy you know, everyday work of the editor, we don't have a lot of time to spend on this. So we would maybe spend 5-10 minutes. And there's a distinction between basically abstract only inquiries or full manuscript inquiries. But anyway, it's kind of a non-committal way, I guess, in order to ping the editor and find out. And probably many times it's about, is this really something you would consider at the journal? Is it in the scope of the journal? In our case, I'm at EMBO Journal. I cover more basic cancer biology. We have a sister journal. This is EMBO Molecular Medicine. They cover more translational journals. So many of the pre-submission inquiries I receive, they are like, this is somewhere between preclinical and clinical work. And would you see this really? And authors are just not sure, right? Is it more for EMBO Journal? Is it more for the other one? And then we can kind of give input. But yeah, I think mostly we would prefer probably either having a longer discussion with the author anyway to get a better grasp, or getting the formal submission. Anyway, we try to be quick. So if I get a pre-submission inquiry, we would like to be back to the author in a day or two.
Okay. Great. Either of the Sarahs?
Sure, I can go quickly. I don't have much to add. I think they're really useful if it's a question of scope. But if it's a question of advance or anything like that, like really it's better to send the full paper. So for Cell Reports Medicine, I found them a little bit more useful. So I was at Cell Reports before, and then it really, if it's biology and it's not purely clinical, then yes, it's in scope. So go ahead and submit. So I didn't really find them as useful. We'll look at them quickly. We're not going to read the full paper at the pre-submission stage because we're always going to prioritize the papers that are actually submitted to the journal. Cell Reports Medicine is where I've seen them be more useful because it is a new journal and people are trying to figure out what our scope is. And so then they can ask us and we can tell you. So that's where it is useful. And, you know, of course, if your work is relevant to a lot of journals and you want to know if they're broadly interested in the topic, then sure, you can query multiple journals at one time. So I can see the appeal. And we also like to look at them quickly. We usually get back to you same day, but they're really only going to take a few minutes of our time. Whereas if it's anything beyond that, I'd suggest submitting the paper because then we'll read your full paper, look at the data, try and understand it a bit better. And we still tend to be fast. So usually if you submit the full paper, we're going to get back to you within a few days.
Great. And Sarah?
Right. Yeah. Yeah, so for us, it's a bit different, I guess, that I can see how when you're submitting a research paper, how sometimes it's difficult to know how, whether it fits without the full paper and the data and the research that's in there. But for us, I think we actually prefer, if somebody's interested in writing a review type article, to send us what we call a proposal, which is essentially a pre-submission inquiry. So same idea, and to submit that on our website. So we have the basic details. And the reason we do that is we just want what that should contain is essentially, maybe an extended abstract. So explaining the rationale for this paper that you want to write, this review article. Why do you want to write it? Why is it different? Why is it timely? All the things I said before. Outline the main sections you want to cover. Give me a list of key references that you'd want to discuss in each section. And that allows us to get a really quick snapshot view of what we're talking about and what the review is going to cover and whether we could actually take it forward or not. And we might look at it and say, okay, yeah, this is great, but actually these bits here, maybe we don't need that because that was covered in something else. If you wrote this, it would be 10,000 words long, and that's longer than we can take. So maybe you should cut it down and think about the scope a little bit. And we give that kind of advice to every author we have. So if you've written the whole thing already and written your 10,000 words, then it all of a sudden becomes an issue of cutting it, and you've spent all this time doing it, and then we have to take it out. So it's almost better for us to send us that sort of synopsis-y stage, very brief outline that we can address those issues up front before you spend time writing it. So that's why that's a little bit more ideal for us from our perspective.